Tomorrow, we had planned a session at noon, which was going to be questions and kind of going over former, at least board type of questions that had a physiologic bent to them. Unfortunately, I'm going to have to cancel on tomorrow unexpectedly, and but I don't know if and when it's going to be rescheduled or not, the question and answer sessions, but you should have gotten in your email or on your site, I don't know how you get them, the PowerPoint files for the first two renal physiology reviews that I gave. Today's is already on there, although I've modified it a bit from what I put on. I kind of went through and changed the order of some things. And then you should also have in that material a set of questions that are kind of like board questions used to be for renal physiology. It's meant to be a review of renal physiology, particularly as it relates to nephrology and renal disease. So there will also be, after you've had a time to look at the questions on your own, we'll also provide an answer key that explains some of the answers that have been gotten. So it can be helpful for those of you that wish to review your renal physiology. I'm going to take up today right where I left off the last time, just overlap a little bit so we keep our orientation. And first picture I'm going to show you, I showed last time, which is of Steve Hebert, a fellow here at UAB from 1973 to 1975. And a point I want to make in the next half hour when I talk about these more distal regions of the segments is how many people who were fellows in this division formerly made enormous contributions in this field. And some of them, including Steve Hebert, really didn't think they were going to go on to be an academic nephrologist. They weren't sure about this research thing, but they made enormous contributions. Steve was on the faculty, then here, went to head nephrology at Vanderbilt, then professor and chair of physiology at Yale, where he died tragically and unexpectedly. He was a Homer Smith Award winner. He was a Berliner Award winner. He was a member of the National Academy of Science. I mean, this guy has got all of the accolades. And as I told you last time, what he's particularly known for is having identified, cloned, and sequenced some of the most important transporters and receptors in the distal part of the nephron. In particular, his early work was in the regulation of salt and water transport in the thick ascending limb and a cortical collecting duct, and by means of systemic hormones more at that time. He then cloned and sequenced the bumetanide or furosemide sensitive NAK2Cl co-transporter in the thick ascending limb, which is about where we were talking about last time. The ROMK channel in the lumen of the thick ascending limb. The thiazide sensitive NaCl, TSC means thiazide sensitive co-transporter in the distal convoluted tubule, and a calcium receptor that we'll see is associated with regulating salt reabsorption in the thick ascending limb. Now, last slide I showed you, I just started and then decided we just didn't have enough time, is about Bartter syndrome. But this slide demonstrates nicely all over again the mechanics of the thick ascending limb cell. And remember the point I was trying to get you to appreciate, that kind of the basic mechanism, forget all these arrows and whatever, that's going on all along the nephron, is sodium is somehow entering passively down this luminal membrane of the epithelial cells, being actively transported out by the sodium-potassium ATPase, which is ubiquitous, but present only on the basolateral membrane. Then you throw in other transporters to design then the operation of this nephron segment the way that you want. We have an unusual transporter, a much more complicated transporter than you see most places, to allow the sodium to enter here. And here it's done in combination with 2 chlorides and a potassium, so it's electrically neutral. And this then is driven by the sodium-potassium ATPase, keeping the sodium out. Chloride can go across the basolateral membrane through chloride-selective channels called cliques, chloride channel, ClC4 in this case, and potassium channels. Potassium can go either way, but here in the thick ascending limb, a lot of it is recirculating, just going in the pump and back out again through this highly permeable so-called ROMK channel for renal outer medullary channel. And I don't think I have it here. Oops, I think I have it.
Let's get escape.
Egg.
Show. Next year.
Is that exit?
Next is fine, yeah.
Okay. Yeah. What happens in Barter's syndrome, and by the way, remember that this ROMK channel predisposed this luminal membrane to be very potassium permeable and to have a lumen positive voltage. And that this lumen positive voltage, the only place along the nephron, gave you a nice driving force for the passive reabsorption of some of the sodium, but importantly also the divalent cations, particularly important in the thick ascending limb. What happens in Barter's syndrome is that there's a loss of function mutation in the NAK2Cl cotransporter. It doesn't completely knock it out in most individuals, but it runs at a very much reduced rate. And these individuals, as you might expect, if you lose this major reabsorptive site for sodium, carries about 25% of the filtered sodium, reabsorbs it, you're going to have salt wasting. And that salt wasting is often accompanied then by hypotension. And it's not due to a lack of function of the afferent arterial or the zona glomerulosa of the adrenal, because they actually become hypertrophied and granular. So there's also hyperaldosteronism that you can measure in the plasma, meaning there's no absence of aldosterone or of renin in the system. It's accompanied by hypokalemia, hypomagnesemia, and I don't know why clinically they always turn things this way and say hypercalciuria, but think of it as hypocalcemia, if you will. Okay, so there's been both wasting of calcium and magnesium. They and sodium are being lost in the urine because this transport mechanism isn't working. Classically, it appears in early childhood and is autosomal recessive. This is the classical form of Barter's syndrome. So just knocking out this pump, lose the positive voltage, you don't reabsorb the sodium very well, you can't reabsorb as much of the calcium magnesium so that there's wasting, and the salt loss is associated with hypotension. So, I should say, yeah, missed a major point here. There is not only a mutation in the NAK2Cl cotransporter, that's the most common, but you can also have Barter's syndrome that's produced by 2 other hits, if you will, or mutations. One is a loss of function mutation in the potassium channel. The other is a loss of function mutation in the chloride channel. And you can see knocking out any of these 3 is just going to gum up the works. You're not going to be able to let this transport cycle, develop a lumen positive voltage, and reabsorb the sodium as effectively. And by the way, all of these mutations involved in Barter's syndrome were identified genetically by Steve Hebert, and was an important translation, if you will, of his work from the basic science to the clinically important. Another important thing that Steve Hebert discovered, largely from work he was doing at Mount Desert with fish, and fish have a very tight regulation of an NAK2Cl cotransporter in their gut. I'm not going to go into why. But from that, Steve, with Brown from Harvard, cloned a calcium receptor that was localized to the basolateral membrane. And with Brown then, they found that this was exactly the same, or nearly the same calcium receptor as you have present on the parathyroid gland. What was it doing here in the thick ascending limb of the loop of Henle? Well, we know more about the mechanism now, but in a simplified view of it, what's happening is occupancy of this receptor by calcium tends to turn down the NAK2Cl cotransporter. And it turns out, very interestingly then, that Steve identified a mutation in the calcium receptor that's associated with, let me see, do I have it back here? No. That's associated with a condition, or localizes to a condition of familial benign hypercalcemia, hypocalciuria syndrome with elevated PTH. So what's happening in these individuals is they've got no break on their NAK2Cl cotransporter, at least no break from the calcium receptor. So this is running wide open, and they're going to get hypercalcemia, hypocalciuria, or hypercalcemia, if you will, due to enhanced reabsorption of the divalents, and you're going to have elevated PTH because you don't have a normal calcium receptor on the parathyroid gland. So all of this fitting very nicely together. Of course, this is a very rare syndrome, but it does point to the importance of possibly also magnesium in regulating the rate of transport of the NAK2Cl cotransporter. Going to move on now to the distal convoluted tubule. And the distal convoluted tubule is characterized by a little different sodium transporter on that luminal membrane, a thiazide-sensitive NaCl cotransporter, just the two ions together. But an important thing about the distal convoluted tubule, it's a very important site of regulated calcium reabsorption, which I'll come to in a moment. And also, from the standpoint of hereditary diseases, it's the location of the defect that's involved in another variant of Barter's syndrome, referred to as the Gittleman's variant. Okay, early distal convoluted tubule. I mean, this is really highly schematic. It's obviously, in proportion, much longer than this. But I want to point out here, primarily just where it is. It begins right after the macula densa. So you remember that the fluid that normally comes here, unless you're taking a loop diuretic or something, is a very low sodium chloride concentration. It has an osmolality of about a third of the plasma, about 100 milliosmoles per liter. So it's usually very dilute in a low sodium chloride concentration. Now it comes into the early distal convoluted tubule, and it turns out that this segment right here, the classical or early distal convoluted tubule, depending on how you want to call it, depending on whether you're into histology or you're into renal physiology, this region is water impermeant, just like the ascending limb of the loop of Henle. And what we have here now, again, the typical pattern of sodium reabsorption. passive entry across the luminal membrane, but in this case, it happens to be coupled to chloride movement in. And then you've got the sodium-potassium ATPase, always present there in the basolateral membrane. And you've got a potassium channel in the basolateral membrane as well. Now, it turns out that the voltage of the lumen here is slightly negative, and it appears to be due to the presence of some ROM-K channel in this distal convoluted portion. However, it's not present nearly as much as in the thick ascending limb, nor in the collecting duct that we'll see later. But there can be a little potassium secretion here that goes on along with the electroneutral sodium chloride reabsorption. Now, the importance of this particularly unique transporter, the NaCl cotransporter, is this is the site of action of the thiazide diuretics. Okay, so that's where they're going to knock this out. Now, when we talk about the Gittleman variant of Bartter syndrome, it involves, instead of the possible 3 loss of function mutations you had in the thick ascending limb, it appears to be, and this was again work by Rick Lifton at Yale with Steve Hebert, was shown to be a autosomal recessive trait that resulted in a functional knockout of the thiazide-sensitive cotransporter. Presents in late childhood or as an adult, typically a little later than classical Barters. It also results in salt wasting, and it also results in potassium wasting here. But as opposed to the classical Barters, the big difference is in the calcium. Instead of being hypercalciuric or hypocalcemic, which I prefer to think it is, but hypercalcuric, as in the regular classical Bartter syndrome. In this case, you're hypocalciuric, and the main reason is that the calcium reabsorption is stimulated in the distal tubule. And this is a finding that's, you're going to see it in other places, but it's very difficult to explain. Pardon me. I'm not going to try to go through the explanations right now because I think it's uncertain. But functionally, what appears to happen is that whenever you inhibit the thiazide cotransporter, you produce more calcium reabsorption. So in other words, that's what happens. You use a thiazide diuretic. For example, when you want to counteract hypocalcemia, hypercalciuria, reabsorb more calcium, you can do that by blocking this. Now, maybe it's because they're both competing for energy from the sodium-potassium ATPase. It may be due to the shifting of where the calcium is being reabsorbed from one region of the nephron to the other. But I'm going to leave it at that and not try to explain it for you, but just try for you to recognize the fact that you kind of have a reciprocal relation between calcium and sodium chloride reabsorption in the distal convoluted tubule. Okay, now a lot of what we know about the distal convoluted tubule and the mechanism of calcium reabsorption is also attributed to a former fellow here at UAB. He was here in a NATO fellowship, 1986 to 1988. He's since become chair of the Department of Physiology, where he had come from originally to UAB as Nijmegen Medical Center. He has received the major renal awards the Gottschalk Award of American Physiological Society, the Pitts Award of the International Society of Physiology, the Homer Smith Award of the American Heart Association and the American Society of Nephrology. And he's also a terrific guy. He has explained a lot of what we know now of the physiology of renal and intestinal calcium and magnesium reabsorption. And I should say, although he started out largely in renal, and that's what he was doing here at the NRTC, he's become even, let's say, more widely recognized for his work in divalent reabsorption in the gut. His biggest claim to fame is that he cloned and sequenced what people thought or hypothesized for a long time had to be there, which was a calcium channel, okay? But he was able finally to clone and sequence one from the gut, the so-called TRPV5 channel in 2000. Since he's cloned an epithelial calcium channel that's present in the distal convoluted tubule, also known as TRPV6. And so he's put together large pieces of the puzzle of calcium, and particularly from a renal standpoint in the distal convoluted tubule. And in many ways, the reabsorption of calcium, where it occurs actively, looks in some ways like that of sodium. First of all, there's passive entry down an electrochemical potential gradient because calcium concentration inside the cell, as you know, is extremely low. So there's a big gradient for the passive entry, and it turns out that there's two active transporters on the basolateral membrane. One is an ATPase. It's a transporter, ATP dependent, so it's a primary active transporter that transports calcium out of the distal convoluted tubule cells, and also some other cells along the nephron, but we'll focus on the distal convoluted. There's also a mechanism of actually using sodium entry down its electrochemical potential gradient, but using 3 sodiums for 1 calcium, using this exchanger to actively transport the calcium out. Now, in order to get these mechanisms to work, remembering that the intracellular calcium concentration is very low, you got to have some way to get the calcium to these calcium binding sites on the active transporters. And it turns out that's very dependent on kind of a network of organized cytoskeleton that binds a calcium binding protein called calbindin. And it happens then that along the nephron, the only places where all of these things come together, this epithelial calcium channel, the calbindin, and the 2 mechanisms of active transport are on the distal convoluted tubule. And in addition, in the distal convoluted tubule, there's receptors for vitamin D3, which enhance the reabsorption of calcium. And there's also here a PTH receptor that can turn up the calcium reabsorption. So you've got all the machinery there. I'm going to leave now the distal convoluted tubule for this obviously much more glamorous cortical collecting duct. I say that because I've worked with it a lot of my life. But the kidney collecting duct is shown here in Technicolor because it's been labeled with different fluorochromes and looked at under a confocal microscopy. And the first thing you notice, one thing you can notice if you look at this carefully, is that there's 2 different cell types. There's these ones here They're labeled with yellow on the luminal membrane. They appear to be the majority. And the reason they're labeling is that we've got a fluorescent antibody to aquaporin 2. Remember, aquaporin 1 was a water channel that's present in the proximal tubule. And aquaporin 2 is similar to aquaporin 1, but it's a different water transporter, one that can be regulated, and we'll talk about that in a moment, and is present in the luminal membrane of these principal cells, as they're called, because they're the majority cell in the collecting duct. But in addition, you've got these other cells in here where the luminal membrane is labeled with blue, no indication of green label, and the blue is an antibody against what's referred to as a VATPase, a vacuolar ATPase. It's a kind of ATPase that you find in mitochondria, for example, and it's that kind of ATPase that can hydrolyze ATP to actively transport hydrogen ions. And as we'll find out in a few minutes, that's very important to have this active transporter to acidify the urine as much as it can be acidified in the collecting duct. So it's active hydrogen ion transporter that occurs here. In addition, there's a fourth red label or orange here, which is an antibody to aquaporin 4, which is another water channel. But this water channel is not present in the luminal membrane like the aquaporin 2 is present only in the basolateral membrane. And as we'll see in a moment, this is what lets the water get out of the cell after it gets in through the aquaporin 2, lets it get out to the medium. By the way, these cells that have the ability to actively transport hydrogen ions are referred to as the intercalated cells. Intercalated meaning they're just kind of stuck in between the principal cells. So, an important thing about and what I'm including here is the connecting tubule. And again, the histological nomenclature can differ here. But it's that segment of the nephron between the distal convoluted tubule, what we call the classical distal convoluted tubule, and is a tubule that connects via these long arcades in the case of deep-seated juxtamedullary nephrons, etc. So it's that segment and the collecting duct system, which kind of occurs, begins a little bit before more than one nephron come together to form a collecting duct. So, when you lump it together this way anatomically, that is from the end of the early distal convoluted tubule to the ducts of Bellini and the medulla, the thing that you find, just grouping it together this way, is this the only nephron region which has receptors for antidiuretic hormone, and it has aquaporin 2, 3, and 4 water channels. The magnitude of the sodium reabsorption is different in these different nephron segments. However, in all of these segments, there's receptors for aldosterone, and in these connecting tubule cells and principal cells, aldosterone stimulates sodium reabsorption, and it does so in part by stimulating the activity of the epithelial sodium channel that I'll come to in a moment. So the important thing here is lumping these regions of the nephron together by virtue of the fact that they all have the ADH receptors and the ability to respond to ADH. So they got to be the area that responds to different hydration needs in the body. And the other is that they're the only region that respond to aldosterone. So, what does vasopressin do in, I'm sure this is familiar to everyone here, but in this region of the nephron, in both the CNT, that's the connecting tubule cells, and the principal cells, is that you have the ability of vasopressin to bind with adenylate cyclase, coupled GS protein, to activate adenylate cyclase with the intracellular production of cyclic AMP. That cyclic AMP production is associated with, and we don't know completely how right now, but changes in the cytoskeleton in this subapical region that causes the insertion of these kind of cigar-shaped vesicles to fuse with the apical membrane and put water channels. These studs looking here on the outside are the aquaporin 2 water channels. And you think, wow, this is a complicated mechanism. You know, you got to shuttle aquaporin 2 water channels all the way from inside the cell, stick them in the membrane. You know, that must take a while. Well, it turns out these are all ready to go, and from the time you add vasopressin to the time you begin to see an antidiuresis, it's on the order of about 15 or 20 minutes. So it's pretty quick. And same thing happens if you take away vasopressin. These channels are retrieved from the membrane in the same kind of time frame. So ADH regulation of water reabsorption in the distal nephron is very rapid, okay, can occur very quickly despite the fact it seems so cumbersome. I now want to shift, because I'm going to combine the two, talking about sodium and water, but I want to shift for a moment here to considering salt reabsorption along the nephron. And just here, for a comparison, the point I want to make here is that you only deliver less than 1% of the salt that was filtered at the glomerulus comes here to the macula densa, 10%, I misspoke, okay? When you consider the aldosterone responsive portion of the nephron, after you've had here the thiazide sensitive cotransport, etc., they absorb a lot of salt. What is delivered to the aldosterone responsive part of the nephron is only about 0.5%, or about 2% a day, 500 millimoles per day. How much is reabsorbed here, and this is now this small remaining fraction, depends on antidiuretic hormone. I'm sorry, depends on aldosterone. And aldosterone, of course, will stimulate the reabsorption, and you can get the sodium excretion down to approaching 0 per day. On the other hand, if you have no aldosterone present at all, okay, aldosterone is completely out of the system, you still end up reabsorbing about half of what comes to this region of the nephron. So you'd excrete about 250 millimoles per day in the complete absence of aldosterone. But as you know, with someone with Addison's, for example, wasting that amount of salt per day, you got to figure, is more than a couple of times the usual daily intake of salt. So it's significant. It's going to cause volume depletion. Now, considering the connecting tubule and the collecting duct here, this is what is present in the way of transporters in the principal cells and in the connecting tubule cells. The mechanism of sodium entry now, again, the overall plan is the same. Passive sodium entry here, actively being transported out by the sodium potassium ATPase there. So what you have now for the entry mechanism is sodium all by itself. Just transports sodium, and it's what's referred to as an epithelial sodium channel. So the sodium, because it enters by itself, it does kind of like the same thing as when you activate sodium channels in nerve or in muscle. It's going to depolarize this membrane. Even though you got a potassium conductance here, the sodium conductance is much more, and it makes the lumen negative. Much, much more negative than in any other region of the nephron. And that lumen negativity gives you a driving force for potassium to be secreted through these ROMK channels that are also present in the cortical collecting tubule. So you got the driving force there. There's some question whether with aldosterone action you increase the number of channels, but we've calculated you don't have to increase the number of potassium channels with aldosterone. Simply the fact you get a bigger lumen negative voltage and enhancement of sodium potassium ATPase is enough to do the job. So, the person who cloned the sodium channel was also a fellow here at UAB, 1987 to 1990, here in nephrology. She then went as a visiting scientist to University of Lausanne, and that's where she actually cloned the sodium channels in collaboration with Renal laboratory. So she cloned and sequenced. Turns out there's 3 subunits of the sodium channel. She cloned and sequenced them, and a tremendous amount of work has come since. Just to give you a rough idea of what the transporter looks like, it's 3 different subunits that appear to be arranged in a 6-pack of 2 types, 2 of each type. But all 3 types are basically the same structure: 2 transmembrane units, a big extracellular loop, and a short intracellular and C-terminal regions. That'll become important when I talk about some mutations in these transporters. That's the only reason I'm showing you this molecular diagram. What does aldosterone do? Well, it's been shown that aldosterone operating through, and remember, the only region of the nephron that has them in around-corticoid receptors, and as we'll see later, the 11-beta-hydroxysteroid dehydrogenase, which is critical for the action. Aldosterone is only present, the receptors, in the connecting tubule and in the collecting duct. There, aldosterone combines with some cortical receptors, migrates to nuclear receptors, and activates a large gene cascade that results in, ultimately, the synthesis and formation of new sodium-potassium ATPase, coupled with a big amplification of the basolateral membrane surface area. So you got more surface area and more pump. Activates the sodium and increases the number of the actual sodium channels. That either directly or indirectly also stimulates potassium secretion. And finally, these this receptor cascade also activates Krebs cycle enzymes to provide more energy for the additional ATP. So citrate synthase in particular is what's markedly enhanced. We know that there's a blocker of this system, an antihypertensive. It's not as potent as many antihypertensives because you're operating only on that smaller amount of sodium that's reabsorbed in this aldosterone responsive region of the nephron. So aldactone or spironolactone can competitively inhibit the aldosterone receptor. Again, the action of aldosterone, you can see it kind of dramatically. In this case, DOAC, deoxycorticosterone acetate, is being used as a surrogate and high levels of DOAC. This, you see a non-DOAC treated cortical collecting duct. This is an intercalated cell. This is a principal cell here. And you see what happens if you add DOAC. There's a big amplification of the basolateral membrane surface area. If you look at it closely there, you can see loads of interdigitations. So you get more than a 2-fold stimulation of the basolateral membrane surface area. And if you stain this for sodium potassium ATPase, it's loaded. It's really increased the amount of sodium potassium ATPase. So we refer to this as the aldosterone responsive distal nephron. It has this epithelial sodium channel. It's a site of action of aldosterone, of ADH, and the water channels are there. This is where ADH regulation occurs. And we've also considered the magnitude of salt reabsorption in different regions of the nephron. And what I want to get to now is, first of all, just the overall feedback mechanism involved in ADH action, and then to give you explanations for some syndromes of where there is defective regulation of distal sodium reabsorption and either volume retention or volume loss. I'd also like to point out, although I don't have time to cover it right now, I've only talked so far about the mechanisms of cation reabsorption in the collecting duct, the aldosterone responsive distal nephron. Let me say that with regard to the principal cells, it appears that almost all the chloride is going between the cells through a paracellular pathway. However, in the intercalated cells, type A intercalated cell, type B intercalated cell, it has now been shown that there is an additional chloride transporter. It has two components, a chloride bicarbonate exchanger and a chloride sodium bicarbonate exchanger. And you will hear more about pendrin, and I just don't have time to get into it now because it does overlap a lot, I think, also with what Mark Bevins will tell you and how it fits into acid base. Okay. Oh, I was just indicating there, type B cell, intercalated cell, actually has a mechanism of hydrogen ion secretion that involves potassium reabsorption, and that there's also the pendrin I'd already spoken about. Let's get on now to the vasopressin. Vasopressin is released from nerve endings in the posterior pituitary, but the ADH is synthesized in two regions of the hypothalamus, primarily the supraoptic and the paraventricular nucleus, but it's stored in the nerve endings in the posterior pituitary, just brought down here by axoplasmic streaming. When an action potential arrives at these nerve endings in the posterior pituitary, you activate calcium channels, and like a lot of secretory processes, the stimulus is calcium entry, which causes fusion of these vesicles containing the antidiuretic hormone, releasing it into the system, which then results in water reabsorption. It causes the activation of the aquaporin 2 water channels. So what releases the antidiuretic hormone? This would be the plasma level. The number one thing, the usual control, is plasma osmolality. So it'll vary from individual to individual, but in a range of about 270 or so, you begin to see detectable plasma ADH. As plasma osmolality rises higher, you then get a rise until you level off at about 20 at osmolalities above about 300. In addition, the body's got another protection, always an important one to consider. That is the thirst mechanism, that not only is your kidney going to work to retain water, but your body is also going to work to try to ingest more water. And when you get into problems where osmoregulation is messed up, it's usually because both mechanisms are being interfered with, okay? Because you can always get around it if you stop drinking or drink, you know, and so you generally have to look a little bit more thoroughly. In addition to what I already mentioned, releasing ADH, it can also be released, as we all know, by a large amount of extracellular volume depletion. Okay, that can serve as a potent and overriding stimulus. But in addition, we also have to consider the possibility of functional hypovolemia. That is where the effective circulating arterial blood volume, effective arterial blood volume is diminished. Those are in the edematous states, and in these situations, body can be massively volume expanded, and yet antidiuretic hormone is being produced. And everybody, I'm sure, is fully familiar with that. The RAS system is also activated, resulting in salt and water retention. Examples would be congestive heart failure, other edematous states, nephrotic syndrome, and cirrhosis. Also AV shunting due to a fistula or congenital defect that gives an effective reduced cardiac output. Again, considering where the sodium is being reabsorbed, it's being reabsorbed primarily in the proximal nephron, but remember, it's this small remaining amount that's being adjusted by the aldosterone responsive distal nephron, and that aldosterone is a prime mover right here. What I want to consider now is examples of where we can show that either hyper or hypotension can occur as a consequence of hypo or hypervolemia due strictly to something that's going on in the kidney. So we can relate to hypertension. Obviously, many other pathways for the genesis of hyper or hypotension, but consider now, important things from the renal standpoint. You can have hypertension, hypertension due to sodium retention. For example, with low plasma renin, just secondary to volume expansion, typically occurs with hypokalemia due to renal potassium wasting and variably alkalosis with a syndrome of hypertension such as this. How can this type of renal hypertension develop? You can think of it as developing due to a problem on either the luminal or the apical side of the collecting duct cell, the principal or connecting tubule cell. We know that mineralocorticoids will drive the sodium reabsorption, and if there is an elevation of mineralocorticoid for one reason or another, it's going to drive an increased sodium reabsorption and an increased potassium secretion. So increased sodium, that'll depolarize the cell more, that'll enhance potassium reabsorption. And examples of where this happened: primary aldosteronism, so-called glucocorticoid remediable aldosteronism, where there's a transposition of the regulatory gene elements between the glucocorticoid and the corticoid receptor, and apparent mineralocorticoid excess type 1. What's that? Well, this is due to a loss of function mutation in the gene for the, and I mentioned this before if you remember, the 11-beta-hydroxysteroid dehydrogenase type 2. And what that particular hormone, enzyme does is convert cortisol to cortisone. And it's important because it turns out the mineralocorticoid receptor is not all that specific. It prefers aldosterone, but cortisol can also occupy this. So really, what makes a aldosterone responsive cell like the distal nephron or a sweat duct uniquely receptive to aldosterone and not to the much higher levels of cortisol are not so much the specificity of the mineralocorticoid receptor, but the presence in mineralocorticoid targets of this enzyme that gets rid of the cortisol and permits only the aldo to work. So a loss of function mutation here is going to act the same, because it's preventing the destruction of this cortisol, act the same as hyperaldosteronism. Now let's consider what would happen with a defect in the sodium channel that might produce hypertension with hypokalemia. Okay, if there were a gain of function mutation in the epithelial sodium channel, that would hyperpolarize more channels in the membrane, would hyperpolarize the luminal voltage, and cause more potassium secretion. And it turns out there's a particularly pertinent genetic defect here that illustrates this. The little kindred in Alabama been studied extensively by Rick Lifton and Dave Warnock, but it involves an autosomal dominant mutation in the cytoplasmic C-terminal region of the beta or the gamma ENaC. And that's why I said the configuration of the ENaC was of some importance, because these mutations all occur in this C-terminal region of beta or gamma. And why is that important? Well, it turns out that the way that the ENaC is regulated in the membrane is by being put into or pulled out of the luminal membrane. And when it's pulled out, what pulls it out is a compound called a ubiquitin ligase, MED4. You don't need to really know these details, but it attaches to the specific motif in the C-terminal region of ENaC. And what happens is, once that attaches, it causes the ubiquitination of the channel and degradation by the proteasome mechanism. So, if the mutations are missing this C-terminal tail, they don't get pulled out of the membrane, and they're there all the time. And so you have hyperactivity of the channels. How about syndromes of hypotension due to salt wasting? You can have it with high plasma renin, secondary to salt loss, for example, usually accompanied by hyperkalemia due to decreased renal potassium excretion and acidosis being variably present. If we go back again to the two sides of the cell where this could be happening, could be due to decreased mineralocorticoid action, mineralocorticoid deficit, that would reduce sodium channel activity, depolarize the membrane, decrease potassium secretion. That could be a primary or secondary adrenocorticoid deficiency. It could also be a mineralocorticoid receptor defect, really quite rare, and is referred to as pseudo-hypoaldosteronism type 1, an autosomal dominant defect. On the other hand, if we look at the other side, you can have loss of function mutations in ENaC. They turn out to be a little bit more rare than gain of function mutations, but in this situation, decreased sodium bed channel activity would depolarize, lead to less potassium secretion, therefore hyperkalemia. And these are found to be due to loss of function mutations in ENaC that occur in the N-terminal region of all three of the subunits. It's referred to as autosomal recessive pseudo-hypoaldosteronism type 1. A lot to remember. What I was trying to show you here, and I'm going to conclude very quickly right now because we're running over time, is that when you look at these common, or not so common in terms of mutations, but then these easily explainable changes in renal salt and water handling, what you seem to see is whenever you have a situation that stimulates the sodium channel, you at the same time, because you depolarize the membrane, of course, more potassium secretion. So the question is the distal nephron always poised to kind of have just a merry-go-round here where more sodium you reabsorb, the more potassium you secrete? And it turns out the answer to this, and we don't know the whole regulation yet, is primarily how easily do you let chloride pass through these junctions? Because the more easily the chloride can go with the sodium that's going through its channel, the less you'll depolarize the membrane, and the less potassium you'll secrete. Okay? So, if you had a mutation that would allow high chloride permeability here in the junctional complexes, sodium and chloride reabsorption could be greatly enhanced. At the same time, you are cutting off potassium secretion. And what that comes to is another condition, which is familial hyperkalemic hypertension. So instead of being hypokalemics, like most of your cases such as this, it's familial hyperkalemic hypertension, or Gordon syndrome, or this very confusing name I find. It's characterized by hypertension with hyperkalemia, acidosis variably present, and it is due to autosomal dominance on 3 different chromosomes that regulate a process that goes beyond what we have time to discuss right now. So let me end it here with this kind of bizarre example. I mean, you're never going to see it, but it's kind of illustrative of, yeah, most of the time you get hypersalt absorption, you get hyperpotassium secretion, and vice versa. But this is a situation where they go in opposite directions. Okay?
Be on the boards though.
Obscure as it is.
Board yesterday.
I saw the word board.
Well,
Thank you.
I'll end it there. Thank you.